Thank you. All right. Thank you for the invitation, and I'm excited to tell you about what my lab thinks. We reflect upon the statistics, which capture the amount of cells that we make and lose every day. And very important for this process are components such as stem cells, these depicted circles here, as well as the environment, the square where they reside, which we refer as niche. And while we understand that these components are key to sustain tissue regeneration, as well as they go awry in processes such as cancer, we still fail to understand what are the intermediate steps that these components take in order to lead to these processes. So in order to tackle this question, my lab, when we started 5.5 years ago, started to ask what are the behaviors and the fate choices that these cells make. What's the role of the stem cells and the niche? In other words, if we were to eliminate them, would we impact tissue regeneration? Third, how is the stem cell pool maintained over time? And lastly, can we learn from normal tissue regeneration, understand the principle of cancer both towards its growth as well as its regression? So in order to start to address the first question, the challenge is that these processes are highly dynamic. And we turn to skin because it's so highly accessible that allows us to develop a new technology that allows us to capture these processes. Of course, we took advantage of decades of work made by giants present in these rooms. They have defined for us lots of principles of homeostasis in the skin, including the hair follicle, which I'm depicting here in black to show you the epithelium, and in red to show you the mesenchyme. And it's starting with the identification of the bulge stem cells from George Cozzarellis, Henry Sun, and Bob Lavker, as well as the extensive characterization coming from many labs, Elaine Fuchs, Fiona Watt, Angela Cristiano, Sarah Miller, and many others. We understand how this tissue regenerates over time. So in order to start to tackle these behaviors and fate choices, though since they are dynamic processes, we wanted to understand whether we could set up a process that allows us to capture it, such as live imaging. And this credit goes to a very talented postdoc in my lab, Padelis Rompolas, who took advantage of several transgenic mice that were already existing and set up an ability for us to watch in live mice how these cells behave. What you are seeing here is the behavior of nuclei, where we can capture not only proliferation, but also migration. And so using these approaches, Padelis could uncover not only basic mechanism of tissue regeneration, but now as a lab, we can also study the emergence of disease from normal tissue growth. So using these approaches, what we understood is that in addition to previous work from George and Mayumi, as well as my postdoctoral work, is that two populations, a quiescent one called bulge stem cells, coming from George's discoveries and doing a tumor, as well as stem cell progeny just below, are two populations that fuel the process of growth, where the follicle goes from its short form in the rest phase to its long form in its growth phase. And so what we wanted to understand is, though, that there are lots of cells within this compartment, and Padelis wanted to understand whether this group of cells were a group of heterogeneous cells or whether they were equivalent in fate. What does it mean? So if you think about the hair follicle, this group of cells have to do two things. One, have to expand this follicle away from the surface and create an outer layer, which is made by several undifferentiated cells, as well as several differentiated cells inside. And so Padelis' question was, if I were to mark a single cell in different locations within this compartment, would each cell give rise to undifferentiated and differentiated layers? So in order to address this question, Padelis had to set up again an ability that wasn't existing in the field, which is the ability to track the same cells and the same follicle in the same live mice over time. And so what he did towards that, he created an approach that allowed him to see lots of follicles at the same time. What you're seeing here are all green bars. Each one is a follicle that then he can zoom in. And using lineage tracing approach, which color for you cells in a way that is permanently then inherited by the progeny, he can mark a single cell in different position using the variability of these tools, and then go back to the same follicle and ask what each cell would do. And so without showing you the data, what Padelis showed in 2013 is that depending on the position of these cells within this compartment, they cover different functions. So these compartments are in contact with the mesenchyme, and so if you are the farthest away from the mesenchymal dermal papilla, these cells will not contribute to the next cycle of the growth. If you're in the middle intermediate position, they will give rise to the undifferentiated cells outside, and if they're in close contact with the mesenchyme down here, they will give rise to the differentiated cells. So what this establishes is that the stem cell position dictates their fate, and it was something that we have seen also in the gut a year later. So altogether, what Padelli's work shows us is not only that we can track the behaviors for the first time in normal tissue regeneration in live mammal and learn the oriented cell division, as well as downward movement drives the process of growth, but also the stem cell position within their compartment that dictates their fate. The next question that Padellis wanted to understand is what's the functional role of these two components? So what we know from Colin Jahoda's work is that when we take these mesenchymal components and we transplant it in the glabrous epidermis, can induce de novo follicle formation. We also know from other work and my postdoctoral work that there are signals that can trigger this transition from quiescence to growth. So what this established is that the mesenchyme is sufficient to induce hair follicle regeneration. The question is whether it is required. So if we eliminate it, would hair follicle growth be impaired? To address that, Padellis set up the ability of eliminating by laser ablation, specifically this population, by coloring with the transgenic that was already available. So what you're seeing here from left to right is an experiment that we do like fly clones. We take several different follicles. Here you're seeing a cluster of 5 follicles. In green, you're seeing the epithelium, and in red now you're seeing the mesenchyme with the tool that Michael Randall made, LEF1 RFP. And so what Padellis did is then ablate this mesenchymal condensate only in 2 of the follicles and leave the surrounding follicle intact so they could serve as internal positive control. By revisiting the same follicles over time, what you saw is that while the surrounding follicle could go on and grow, the follicles in which the mesenchyme was removed instead, they were stuck and they couldn't go on. So what this established is that, in fact, mesenchyme is also required, not only sufficient, to induce the transition from quiescence to growth. Padellis wanted to ask the converse question and ask whether this specific stem cell population that inhabits the bulge was instead required for the follicle growth. So what he saw is something that was consistent and remarkable, which is that as he was eliminating this population, whether he eliminated the stem cell compartment or the stem cell progeny, these populations were readily compensated for. So you would see that from black, where he shoots these cells and eliminates them, then it is not only the green gets recovered by other cells coming in, but also that the follicles continue to grow. So this is consistent with the work from Mayumi Ito and George Kotsarellis, where we knew that there is a level of plasticity where these follicles can regenerate. But what we wanted to know is what cells compensate for the loss of bulge stem cells. So in order to address this question, what Padellis did is to use time-lapse imaging upon ablation. So eliminate the cells and then perform movies and ask which cells get engaged to repair the hole that was created. And so we saw 2 things. One is consistent with Mayumi data. The proliferation was seen in the adjacent cells close to the gap. And this may be also leftover stem cells, given that the stem cell ablation is not a perfect technique. But what he also noticed is that there is an activity very far away from the site of ablation. This site in particular is called infundibulum. It's several cells away from the site of ablation, where what you are seeing is that these cells are actively proliferating, where down below their bulge was eliminated. And so what this prompted Padellis to have as an hypothesis is that perhaps this cell activity may be functional towards the recovery of that compartment. So he hypothesized the cells far away from the site of ablation can also repair that gap and come in and reconstitute the bulge. To address this question, he had to have a rigorous approach where he could mark these cells distinctively from the bulge population. And so he used, again, CRIS system, lineage system, where he can mark these cells in red and green, and they are permanently marked so they cannot lose the red, and then choose follicles in which instead the compartment he wants to ablate is only colored by one color, the green. And so by following follicles like that and shooting this compartment, it then can ask whether the recovery is done by green cells, which means only the cells that are close by, or red cells, cells that are far away. So if you follow, please, from left to right, what you're seeing is a representative example where Padelis chooses follicle of this kind, shoots the cells, and you're seeing this red halo, which ensures that the ablation was efficient. And what he sees is then recovery that occurs over time of these follicles, whether these follicles are now made by red cells that are coming from very far away. So this was very interesting and novel because it explains the cells that normally would not participate the way at least we understand it today to hair follicle regeneration in the face of injury can come in and do the job for the hair follicle stem cells. So what I've showed you is that then the stem cell bulge ablation shows a high level of plasticity where hair follicle regeneration can occur. And so altogether, what Padelis has shown you is that stem cells are dispensable, or at least this specific group of stem cells, the hair follicle stem cells, the bulge, because other epithelial cells can compensate in function and cover for the role, whereas the mesenchymal dermal papilla niche is required for hair follicle growth. So what we wanted to understand next is how this stem cell pool gets maintained over time. So we were reflecting upon this process of regression that occurs upon completion of the growth, and where the large majority of the cells in the hair follicle have to be eliminated. Strikingly, it can occur within 3 days, and 90% of these cells are lost. And the way we understand it in literature is that there is a massive apoptosis that will lead to the elimination of this lower portion of the follicle. Yet the stem cells will be maintained through this cycle and will be reconstituted at the end of it. So in order to start to tackle this question, a very talented student in my lab, Kai Mesa, decided to tackle this broader question by asking who dies, how are these dead cells removed, and how is the regression initiated? So this is Kai, and what he did is apply the technology we had to start to capture this process of regression. And what you're seeing now is 4 follicles in the regression phase, where we are still labeling nuclei that with this histone fusion with GFP can allow us to capture death. Remarkably, we didn't see a large amount of death. Instead, when Kai started to look at this recording, he realized that death was limited in the outer layer that contains all the undifferentiated cells. This is displayed here in the upper layers, whereas when you focus inside the follicles, you would see that all the differentiated layers would not die, and instead they will keep being displaced upwards like you would see during normal growth. So what he wanted to then ask is, how are these dead cells removed? And his favorite hypothesis was that immune cells would come in and clear these dead bodies. So what he did is cross the same transgenic where we can mark the epithelial cells and the death with the many different combinations that will label immune cells, such as lysine Cre in this case. And so what you're seeing is, again, the epithelium in green and the immune cells in red. And we were expecting to see 2 things, either the red cells coming into the follicle, or even seeing fragments of these epithelial cells within immune cells outside the follicles. And we didn't see either of them. Even when we follow in real time this death, we didn't see the surrounding immune cells in red coming in and taking this debris. So this left Kai with the idea that some other mechanism must be eliminating these dead cells. And so what Kai did is to go back to his recording and observe closely how these dead cells were dying. And what he saw is that these nuclei will fragment. The fragments will remain for a very long time, but also they will get relocated around the neighboring cells. So this prompted Kai to formulate the hypothesis that perhaps epithelial cells may act as phagocytes. And in reality, this may not be coming as a surprise to us, because in the 70s, there were many papers in culture, certainly, where we would see the epithelial cells can engulf beads, as well as they can also take up melanosomes in order to be pigmented. So what Kai did is to, first of all, go to EM and ask, during the regression phase, do we see healthy cells in the epithelium that contain apoptotic bodies? And here, it pseudo-color for you several epithelial cells in the epithelial strand, and you see here in blue and green that there are several apoptotic bodies within cells that are healthy and that are keratinocyte. So what he wanted to ask the next is, can we understand the process in its entirety? Can we follow a cell from the moment that it detaches from the basement membrane, forms the apoptotic bodies, and be engulfed, so that we can understand the kinetics of this process? So in order to do that, he turned to a new combination of transgenic, where now he can follow the membrane outline of the cells and instead mark the nuclei in red. And what he saw is, if you see this tangential view of the follicle, you see that now it's marked to becoming apoptotic, and you can follow all the fragments that are going to be formed. And also you will see these fragments now forming and going inside the follicle and then being taken up by these neighboring cells. You're seeing here a cross view of the follicle to show you perhaps better how these apoptotic bodies are engulfed within the cells. So in order for visualizing this maybe perhaps more efficiently, what Kai did is to highlight the cell that in this case was dying in red and show you that there are 3 apoptotic bodies in this case being formed, and you're seeing these 3 tracks. And then it's highlighting for you one of these apoptotic bodies which end up in these 1 neighboring cells. But the other apoptotic bodies are also ending up in other epithelial cells, which elucidate the mechanism by which there is a collective burden elimination of this apoptotic body by several different cells. So what Kai sees is then in essence that these differentiated cells are protected from death, that these undifferentiated cells are targeted for death, and their apoptotic debris are internalized by neighboring cells. So to answer then who dies and how are dead cells clear, what Kai was able to demonstrate is that the basal epithelial cells act as phagocytes and collectively remove their apoptotic neighbors. So when he wanted to address how is the regression initiated, Kai thought that perhaps by doing time lapses during regression in a time course, it could understand how cell death is regulated and whether it is spatially regulated. And so he asked whether cell death is spatially regulated by then doing time lapses and marking the position of the death, in this case as a cross, with respect to the follicle. And what he noticed is that at the beginning of the regression phase, this death was mostly seen close to the mesenchyme. This came as a surprise to him because normally in literature, in limb development in other scenarios, we know about the mesenchymal to epithelial interaction as a regulator of growth in a positive manner. Here instead, this stimulated Kai to think that perhaps mesenchymal crosstalk may be required for regression and therefore epithelial clearance. To test this hypothesis, Kai turned to the approach that Padelis had established to do laser ablation, with the hypothesis that if the mesenchyme is required for this regression phase, then you should see a lingering of these follicles and they should not regress. Instead, if it would not be required, the follicle should shorten and become in the resting phase like its neighbors. And so when he did this experiment from left to right, what he saw is that if he takes this tube, now this is a follicle in regression, all the green cells are epithelial, this is the mesenchyme. If he shoots the mesenchyme of this follicle, it keeps the surrounding intact. When he follows over time, he sees the surrounding follicle shortening. But what he saw is that the follicle that he ablates for the mesenchyme remain long. Interestingly, they remain long, but without differentiated layers. So the differentiated layers could move on and be extruded like they would normally do during regression or growth. So this established the cellular involvement with respect to this process, and so established that the mesenchyme is required for this regression process, but leaves unanswered what signaling may be mediating this phase. So what Kai did in order to understand this question is he started to do search of the literature. He realized that colleagues have shown previously that if you inject ectopic TGF-beta1 ligand, you can induce precocious regression. So the first question he had is whether a TGF-beta is active during the regression phase at the right time in the right place. So he isolated the mesenchymal cells and did RT-PCR as well as staining. And in these slides is the summary of what he saw. So in this cartoon, I'm depicting for you the components we are speaking about. In blue, the mesenchyme, and what is saw by RT-PCR, that the TGF-beta1 ligand gets upregulated at the beginning of the regression phase when compared to growth or rest. Also, when you look at the target genes by RT-PCR in the epithelium, as well as by staining, you saw that TGF-beta is activated very close to the mesenchyme at the beginning of the regression phase. So what this establishes, this pathway is engaged at the right time, at the right place, but it doesn't establish a functional link. So in order to address that, what Kai did is to remove the receptor in the epithelium and ask whether that would lead to a similar phenotype as seen by mesenchymal ablation, and it did. So by removing the TGF-beta receptor 1 into the outer root sheet, or these undifferentiated layers, at the same time that the regression would start, then what it sees is that it has a similar phenotype where all these excess undifferentiated cells remain in the follicle, whereas follicles coming from the flux plus control littermate, they regress like normal follicle would. So what this establishes is that we have uncovered both cellular signaling components important for this process. And what this also stimulated us to think is that here there is a bag of excess undifferentiated cells that remain, that were otherwise doomed and destined to die. And so it prompted Kai to really ask whether this regression process eliminates incompetent cells or it just limits the stem cell niche pool. And so to ask this question, ideally one would want to induce growth upon triggering this phenotype and ask whether these cells are competent for a new growth. We were extremely lucky that Jeff Berninsky published a paper in Dev Cell where he described the mesenchymal dermal papilla cells can actually regenerate during the regression phase. So we are very lucky to observe the same as we were ablating the mesenchymal in these follicles, obtaining this phenotype. What we would see is that, in fact, a new DP would be regenerated at the bottom. So by following these follicles, we could then address whether this is an excess of functional cells or whether these cells, having been destined to die, they would not be able to sustain normal tissue growth. So what you're seeing here on the left is a normal control. On the right is the experiment. On the left, you would see a normal progression of a follicle. And what Kai saw is that in these phenotypic follicles, as the DP was regenerated, these follicles would initiate growth at the tip and then go on into the normal cycle and go back to the rest phase like their neighboring. So this is very provocative in our mind because in the field, we think about stem cell as having a clock and being exhausted after a number of division. Yet this may suggest that it may depend more on the extrinsic regulation, their ability to produce a new growth. And so what this establishes is that this regulation of regression is an extrinsic regulation that has two players, at least according to what we see. One is the mesenchymal, and the other is TGF-beta activation. And also that the niche may be sufficient to maintain tissue homeostasis and even restore a phenotype like the one induced by mesenchymal ablation. So lastly, I would like to address how our understanding of physiology has inspired us to look at cancer models. And this is primarily inspired by a very talented clinician at Yale, Dr. Christine Koh. As was said in the introduction, Christine is an expert in these self-regressing tumors that we found extremely stimulating because they reminded us of the natural cycle of skin growth and regression. And so Janet Sitt, a postdoc in the lab, took these two models, parallel model, a malignant one and benign one, to start to understand processes of growth and regression. So we begin by starting to look at the growth, and the growth of these tumors is fueled by Wnt similar to squamous cell carcinoma. And so what Peggy Myung, a clinician scientist in my lab, as well as Elizabeth, were able to drive is understanding how Wnt within stem cells may drive their behavior. And so they took advantage of the technology in the lab, and then now instead of only marking cells, also delivering the activation of Wnt through a beta-catenin gain of function, and then ask how these beta-catenin cells will clonally expand to make tumors. And so what Peggy and Elizabeth discovered is that, in fact, they do not do so by clonal expansion, but rather by recruiting wild-type cells through a mechanism of upregulation of Wnt. I'm not going to spoil that because Peggy is going to present this work to you in her own talk, but she will show you how she can track the beta-catenin cells in red as well as the wild-type cells in green, and then understanding the dynamics by which cells that bury an oncogenic hit can trigger tumor formation. So what was intriguing for us in these tumors was also the regression phase. And what Gianni did is to map all the molecular aspects that were holding true not only in the mouse model, but also when compared to human. And what Gianni was able to do is to identify not only the upstream, the retinoacid, but also the cascade by which this would occur by switching off Wnt and inducing differentiation and therefore regression. So what Gianni wanted to understand is whether this mechanism and this platform we identify by using these self-regressing tumors could be utilized to induce regression of squamous cell carcinoma. And to do that, they injected tumors, either squamous cell carcinoma or benign tumors, with retinoic acid, and showed that they all can regress, although at different extent. So what you're seeing here is a group of tumors who they were untreated and then measured in size over time, MOC-treated or retinoic acid-treated. And we have highlighted for you the squamous cell carcinoma in red so that you can see their performance over time, as well as the performance of benign keratoacanthoma. So what this established is that this platform of self-regressing tumors can be very powerful in that they can identify knots of the molecular cascade that then we can apply to tumors that do normally regress and make them behave somewhat like these benign tumors. Yet, when we went deep down into the analysis, we realized they do not behave identically. And in fact, while they upregulate the differentiation program, which is the favorite way by which the keratoacanthoma self-regress, they also trigger apoptosis. And so that has spurred for us the desire to understand the mutational landscape that differentiated these two types of tumors. And so this is an ongoing project in the lab where we want to identify the signal that distinguish these malignancies versus benign tumors. And what we are doing, we are taking an exome sequencing combined RNA sequencing approach with Dongwen and Zhenzi Liu, and then using a comparative approach like we have done before in mouse and human, and then tested the hits we find initially in subcutaneous injection so that we can readily test this candidate. And this approach has been already quite efficient, as we have done a pilot screening where we identify 6 genes. And one of them, perlecan, turned out to be very interesting. And Katie Swartz is going to present a poster about that, so I'm not going to spoil it for her. And what we want to do with these hits is also go back to our approach in live imaging and start to capture the emergence of cancer from normal tissue growth. I remind you that we have learned through Padeli's work that depending on the position, these cells can do different things. Now we can go back and use these hits to understand whether we can capture the malignant behavior from the beginning, depending on the position and the cellular function. So with that, what I told you is that Wnt beta-catenin acts by recruiting wild-type cells into this tumor and set an understanding that we can then investigate in malignant tumor, and that also Peggy is going to take with her in her own lab into understanding basal cell carcinoma principles, as well as the retinoic acid, which is this molecular knot we understood from self-regressing tumors, can induce the regression of malignant tumors. And so in conclusion, I just wanted to highlight that my lab has been established a way for us to look at behaviors in real time, as well as establishing the ability to address the functional role for different components and investigate this principle in tumors during growth and regression. And we think that these principles are conserved in other tissue, not only because we have seen publishing that have confirmed our information also in other tissue, but also because the same components as well as the same signaling are conserved in other tissues such as the blood, which is high turnover, as well as low turnover such as the brain. And I wanted to highlight for you Padelis, Peggy, and Katie. There are the 3 of them from my lab at this conference. They're giving 2 talks and a poster, and I hope you can visit them. And also Padelis and Peggy are on their way to establish their own lab, so I'm sure they will be very pleased with your visit. And with that, I would like to finish by acknowledging my team. It's an amazing group of people, very driven and very talented. Our collaborators at Yale, as well as the American Cancer Society, which started to fund us early on, as well as NIMS and NICEF. Thank you.